Hi, and welcome to Holmesglen. My name is Adrian, and I'm currently studying the Diploma of Laboratory Techniques. This semester, I had the opportunity to gain valuable industry and field experience with the Dna Barcoding Project. Let me show you what that entails. Dna barcoding is the process of getting DNA samples from an area and analysing the DNA to see what animals or flora are actually present in that area. And why it's a barcode is because as DNA is, it's unique, so it's as unique as a barcode on the back of the packaging in the supermarket. And why it's novel is because Holmesglen is the only university/TAFE that does DNA barcoding as part of a project for the students. Lake Eildon's never really been profiled for what species of animal or flora there are there. So firstly, in just that respect, we'd be getting a more comprehensive overview of what's in the area, and that will also be published online and held in databases for other people around the world to view. Well, apart from it looks very nice on a resume, and we have published data online at the point as well, so that's also great. It also shows that we can go out into the field, sample, we can do the same kind of work at other places. The molecular biology skills of PCR and genetic extraction is also very helpful in the industry, no matter if we're in molecular biology or in an industry that's just an offshoot of it, it will all be helpful.
So here we're at the Rural Learning Centre near Lake Eildon in Victoria. We're undertaking a problem-based learning approach here where the students are actually undertaking real research under the guise of a barcoding project. The students are now using the idyllic location around the RLC to sample various ecosystems that we have down here, and we can use these unique barcodes to actually identify and profile the different ecological systems here so we can get a gauge of the floral diversity around the Lake Eildon area.
So we're at the Department of Environment and Primary Industries at Snobs Creek, and we've just received our samples of rainbow trout eggs and brown trout eggs, and the way those were taken out were the fish was just taken out of their little pools, and then they were just basically squeezed like a Ziploc bag and just squeezed out at the end of the fish, and then we're put into these bags, and now we're going to go and extract the DNA.
So what we're doing here is taking our 20 samples from our random quadrant, and then we're putting them in our tubes, and what we do is we put the barcode number into an app that Tim has on his iPhone. That's stored along with a photographic picture we take, and so in the future what that will do will allow us to just take a scan of the barcode, which will then show us to what sample it was that we've put in here.
This approach to learning has never been undertaken at any TAFE or university around Victoria. It's beneficial because students are gaining real-world laboratory experience where students are actually undertaking real research. They're learning employability skills that they can take with them to their subsequent jobs, which are used in pretty much every research and development laboratory in Australia are using these techniques. potentially placing our students ahead of most other students with similar qualifications.
Hi, I'm Kim. Earlier this year we went on a Dna Barcoding project. We stayed up at Lake Eildon at the Homestead Rural Learning Centre. It was a great and beautiful facility. It also gave us great practical experience within the work and field, which also gives us confidence that we can go out and get a job in the workplace. It was an awesome experience, and I really believe that it's laid down great foundations for my future.